So here we are. And here's another set of another internship program that happens in the summer that hasn't gotten enough applicants. And it's a STEM internship program. And you can ask me about it if you're interested. This is where we're at. So we have our exam in just less than 2 weeks on the 24th. We have the estrogen receptor mind map assignment due today. We're reading chapter 16 on cell signaling and writing down questions and bringing those questions into class and asking them. And then we have a PGF receptor and wound healing assignment that's due next Tuesday. And that's a bigger assignment. I think the video is 45 minutes long, and people are sending me their notes. So I talked about that last time and in the announcement like that, okay? So we talked last time about the PGF receptor pathway. We have an assignment on that. It's a really important pathway for us. And at the end of class, we zoomed in on the enhancer of a PGF target gene. And it could be MYC. This could be MYC because under a scab, MYC is one of the target genes for the PDGF and the PGF receptor pathway. That's not true in every cell. It varies, but that's a target here. And we found that when we zoomed in on the figure from the book, there's actually a second transcription factor that's needed here and is called SRF for serum response factor. And consequently, the enhancer is called the SRE, the serum response element. And we talked about that last time for a couple of reasons. One is because of how it was discovered and how the scientists used the methods that we learned about in the transcription factor assignment to do a footprinting and gel shift and regulatory region deletion studies and purification and analysis to find this protein. And they have a really good name based on the assay they had for it, which is they would have serum starved cells growing under sterile conditions in the lab, and they would have all the nutrients, all the oxygen they need, but they're just missing those growth factors from that cat serum. And when that got added, things would go forward in the cell cycle. So the scientists said, okay, well, what genes get turned on, and so on and so forth, like that, okay? So that all fits together really well. So we just have one more thing, kind of thing to say about that, and here it is, and that is serum response factor is activated at wound sites primarily through a Rho-actin signaling. And we're not going to learn about that pathway, but it's a different pathway. And so this is a good example of how signal transduction pathways amplify and integrate signals. We need both those signals to get that mix going. And so, in a broad sense, and at the end of this chapter 16, the author talks about what's the purpose of these complicated signal transduction pathways. And the two main things are we're going to amplify that signal, make a big cellular response, probably a variety of different responses. We can have genomic responses, turn on a variety of genes. We're going to have non-genomic responses and have changes in the metabolism or the cytoskeleton and so forth like that, right? And then the other thing is this integration. And people often call that integration checks and balances. And so that sounds great. What's a practical example of that? In real life, so probably every family, whether they're aware of it or not, has a certain type of cancer that runs in their family. Breast cancer, uterine cancer, and so forth, like that. And they might not be aware of it because, it's infrequent, like that, in a lot of cases. And what's going on there is understood to be that family is carrying already a mutation that weakens a check and balance situation. And if they get the mutation in the other one, that's going to really likely, lead to that specific type of cancer. And that helps us see an example like this, how this checks and balances is significant for our health, and how cancer is like a multi-step, multi-hit progression. And these would be specific cancers, again, a type of breast cancer, maybe an ER-positive breast cancer that runs in that family, because they already have allele or alleles that are mutant that already set them up. They're found to be vulnerable to another mutation in that type of case, like that. So we'll learn more about that. Then, so we're not learning about that other pathway. Then I just wanted to show you this, and that is this little overview statement. It says, well, ALK1 is, and they call it an accessory protein that interacts with SRF to form a complex that activates specific genes. That's what we're learning about. It is actually the overall process and activation of SRF that is indispensable for the healing process. So it turns out in our course, we're really focused in on regulating the cell cycle, because that's what usually gets disrupted in cancer. But it turns out wound healing under that scab, it includes more things, more cell responses than just the cell cycle. And that's why they put emphasis on this SRF. We're psyched. We're learning about RAS and we're learning about ALK and that pathway because it's super significant in our normal health in a variety of places. Here, it turns out it's playing a really big role in the cell cycle. But when you look at mutations that are missing SRF, there is not good healing. So in a mouse, in a model, if you just knock out SRF, the animal does not heal well. It's not just a cell cycle issue. And it turns out that when we're talking about this, our elk story especially occurs in the epidermis. But there are wounds that go deep and include epidermis and dermis and even muscle. And SRF plays a big role in those healing of injuries in all those tissues like that. So just be aware of that again, we're not even learning about SRF other than what we learned already. It's not going to come up again in our course except for on exams. But yet it's really important in the overall healing process. And so that's something we have to mention every once in a while. We're not oversimplifying things in our course to introduce these concepts and principles. We really have to target some examples that are memorable and significant, like that, okay? In our overall health and in particular in cancer, okay? Great. So that's that, back to that, and so we're always set with that, okay? So I just wanted to talk about that, and I have did I already show what we're doing this week, I guess? Yeah, okay, so we're good. So we're good to roll on to talking more detail about chapter 16. I want to just pause, and check in with people if they have questions about anything at all. We're already at a point where we can be reviewing for this first exam. And the learning just stacks up. So it's not like you have to go back in the second exam and learn everything again from the first exam. The hope is you'll have lots of it already in hand, like that. Something I just thought of, I would say for the first exam, I would watch those muscle differentiation videos again, because we did that at the start of our course, and I think we really stacked up a lot of understanding and perspective of what's expected in this course, and what I'm trying to explain in those videos. We don't care about muscle differentiation very much. We care about genes and the regulation of transcription of genes by transcription factors, like that. No? Okay? So this is the flow, and I showed this overall flow of this chapter previously. And what I've done here is we'll start adding in names. And so the very first set of receptors that we've learned about are nuclear receptors. And our favorite is estrogen receptor. But we need to know that there are 48 nuclear receptors in a human, and they do a lot of things like that. So we'll learn more about the nuclear receptors, and they'll pop up. And that's the logical place to start, because no signal transduction pathway is needed to activate a nuclear receptor. Okay? And you should be able to explain why that's true. So here we are talking about PGF receptor, and it's a member of the receptor tyrosine kinases, of which there are 58 in our body. So a bunch of different kinds. And we're just going to learn about 2 for this exam. We'll add another one specifically for the second exam. And so we really, as a representative of that, a very important class of receptors, we really want to know that PGF receptor pathway. And so we pick on that example, and we have that assignment of the role of PGF receptor under scab. And that's the assignment that's due for next Tuesday, like that, okay? So we're talking about that, and we'll run into this insulin receptor as well, okay? So the next thing is, before we kind of go further, is, so with the PGF receptor, we've learned about one signal transduction pathway, and it's that one that includes RAS. we'll call it that. It's the RAS signaling pathway. It includes that RAF, MEC, ERK, ELK, like that. So, you know, it includes a lot of players, but, you know, it's that RAS pathway. And when we're talking about this class of receptors, we'll learn about 3 different signal transduction pathways. And that's because these different receptors have multiple signal transduction pathways that they use in different cells, you know, in different contexts like that, okay? So that's our first one. And I showed this before, you know, there's this overview and information about how this receptor pathway works. There's this role of RAS in this specific signal transduction pathway. There's the signal transduction pathway we've learned about. Here are the, what we'll say are, I put them in quotes because they're what we'll call them for our course. And so we have 3 receptor pathways for the tyrosine kinase receptors. And one is called RAS, and one is called PI3 kinase, and one is called phospholipase C. And the way to think about that is RAS is used in this first one and not in those. So each one of these is distinct. Sometimes in a certain cell, one of the receptor tyrosine kinases can actually activate 2 of these pathways, or 2 of another, because there are more than just 3. But we need to learn them and sort of separate them out as we learn about them, okay? And what we're learning about is we're learning about that those signal transduction pathways in that biochemistry. So here's our second one, and our second one is called PI3 kinase, and it's actually the FOXO story that we saw the first day of class. So the very first day of class, or first week of class, we said, look at that FOXO transcription factor. It can't get into the nucleus, ho, ho, ho. It got phosphorylated, that's one way it's regulated. It's blocked from activity because it can't get into the nucleus, and it's held in the cytoplasm by this 14-3-3, like that, okay? All right, and this is actually the insulin receptor, and when we think of this pathway, we shouldn't think too much about diabetes. The insulin receptor pathway plays a role in a lot of different things in our bodies, and people in general refer to the insulin receptor pathway and insulin as the insulin signaling system, because in humans there are 3 paralogs in the insulin family, and there are 5 or 6 paralogs in the insulin receptor pathway. So they do a variety of things like that. So don't get too hung up on this being about diabetes, because it's actually about other things and the role of it. But we want to look at this, and what you can see here is that when this receptor gets activated, and what receptor, it could be insulin and the insulin receptor, and it's not playing its role in overall diabetes, but it's playing a specific role in the cell in our body. So when it does that, it activates this enzyme, PI3 kinase. So it's going to be a kinase. It's going to phosphorylate something. And kinases, of which there are like 600 different ones in humans, kinases phosphorylate specific target molecules, right? It's an enzyme, so it's a very specific substrate. And usually people think of proteins. And this might be the only example in our course where a kinase is phosphorylating a different kind of molecule. And what it's going to phosphorylate is it's going to phosphorylate this lipid. And it's a phospholipid that's on the inner membrane of the lipid bilayer of the cell membrane. And it has a name, and we'll see its name, its long name. But for us, the easiest thing for us to just remember is that this PI3 kinase gets activated by a tyrosine receptor kinase. And it's going to phosphorylate its target molecule, and a target molecule is actually a phospholipid in the phospholipid bilayer. So just like, you've seen, all the time in biology, right? We have a lipid bilayer like that, right? And it's made up of phospholipids, and there are many different kinds of phospholipids, and this is one. And I'll tell you the long name, but we can learn the short name, that's just fine. The short name for this is PIP2, and that's the substrate, and the product is PIP3. like that. And then that's activated. It's going to do something, right? So PIP2 gets phosphorylated to PIP3. PIP3 is active, and then we can look back at this and see what it does. So that PIP3, it stays in the membrane. It's a phospholipid molecule, so it's stuck in the membrane. Phospholipids in the cell membrane and any membrane diffuse really fast, side to side. So in just a minute or two, this PIP3 could float, diffuse, all around the inner cell membrane. They just are moving way too fast for us to even imagine. And so it's diffusing around, and what it does, it becomes the binding site of this protein complex that's called mTOR2, but we don't need to learn about it, just that it's going to activate something. And then this is the one we ought to learn about, is it's going to, that's going to phosphorylate that, and that phosphorylates this AKT. So we want to pay attention to AKT. So for this pathway, we could say, well, the insulin receptor binds insulin, activates PIP3 kinase, phosphorylates PIP2 to PIP3, and then PIP3 binds to a protein that activates AKT by phosphorylating it, and the phosphorylated form of AKT phosphorylates FOXO. So that would be great for us to know about that pathway, because we already know a lot about FOXO, and here's what FOXO is trying to do and stuff like that, right? So that's really what we want to learn about the PI3 kinase pathway, because we're using the PDGF receptor pathway and RAS as our main favorite example. But we need to learn about a variety of these just to demonstrate that there are several different pathways. So we have, with just one receptor in the receptor tyrosine kinase family, we have several different roles it can play in different cells throughout our body or at different times, different conditions, okay? So that's all we need to learn about that one, is to go back to the FOXO story and learn that pathway in that level of detail. So John?
Question, Professor.
Yeah,
You
go.
say PIP3 bind to a protein that activates
AKT.
AK.
Yeah, and it's actually binding. It has an option. It shows that it has an option here. PIP3 can bind to either this mTORC or PDK1, and either of those can phosphorylate AKT. So that's one reason we're not worried about those too. It's just somehow that PIP3 is going to activate AKT, and AKT is going to float around in the cell, phosphorylating FOXO, and blocking it, okay? All right, so this is the first time probably you've ever heard the idea of lipids playing this kind of role, a cell membrane. And so lipids famously have three roles in our bodies, in the biochemistry of our bodies. So that's obesity is storage of types of lipid molecules. There are a lot of different kinds of lipids. Then big part of the cell membrane and any membrane. And then here's what we're focused on, is lipids in cell signaling, as in information pathways. And we've learned about the steroid hormones. They are classified as lipids, highly hydrophobic lipids. And then messenger molecules like this. So this PIP3 is like a cell messenger. It's part of the membrane. It's playing that structural role, being in the membrane, but it's also playing a role in these information pathways. So that's probably something new for you. Here's the structure of it. You don't have to memorize it. Here's the name of it, phosphatidylinositol. So phosphatidylinositol triphosphate is the full name for PIP3, the active form of it. And it gets phosphorylated up here on the head of the phospholipids like that. Yep. So it's, that same structure that you've seen. It's just that we have many different types of phospholipids, and they have these, these different roles they play, right? They can store energy, they can be part of important structures, and they can carry information like this, okay? So that's just that figure again, and great. So, again, we talked a lot about RAS like that, but this is what we need to know, is we need to learn about that pathway too, okay? And that's going to interact with this PIP2, PIP3 story. And that phosphatidylinositol triphosphate is really a famous molecule in our bodies. So we're just introducing that. Yeah,
Can you go back to the PI3? Just so I can take a picture of it. That diagram. I just can't see it.
Oh, this one here?
Thank you.
Yeah.
I just need to take a picture so I can see
Sure,
it.
yeah, absolutely. And that's the figure that we've shown, and it's in your chapter 16, 16.25, like that. Yeah, and it's true, sometimes you wish I zoomed in on those so you could more clearly see that, yeah, yeah. And so I try to do that sometimes, and I just fail to do it there.
So I have a question.
Yep.
Is there any lipid that is not phosphorylated, and what can happen to a lipid if it's not phosphorylated?
Yeah, and so there would be lots and lots of lipids that are not phosphorylated. And this phosphate group is playing this information role. So you see, like it's that PIP2 is already part of the membrane because it's a really traditional phospholipid. And that's where it has these hydrophobic tails that make it stick into the membrane and assemble the membrane. And it has this hydrophilic head. And that hydrophilic head is what's getting phosphorylated. So just imagine, the ceiling of our room, and that's the inner surface of the cell membrane. And floating around up there is like a disco ball, right? And we're going to turn it on, and that's going to get the party started. And we're going to turn it on by phosphorylating it. So it's a physical thing, right? It's just floating there. And there's something, the head of it is hydrophilic, so it's poking out, sticking out into the cytoplasm. It's, why is it using that? And the answer would be, well, the receptor is in the cell membrane. So people call this, they say that the PIP2 is anchored in the cell membrane. That RAS protein is anchored in the cell membrane. So they can diffuse around, but not throughout the cell. And so see this, it also provides kind of a nice visual of that. All of these guys are anchored to the cell membrane, and then it's AKT that's let loose to race around inside the cell, And we'll see that again and again. That was true for RAS and these other pathways. Very often, the first steps in the pathway are anchored, and that means they're moving around, but they're trapped on that inner surface of the cell membrane to concentrate the action there, and then they'll release something. And we'll see on our next signaling pathway that happens big time, okay? Yeah, other questions people have? Okay, so signaling like that. And there that is, right? And that's a figure again, okay? So then the third one we're gonna learn about is called phospholipase C. And so this is an enzyme because of that, ASE. So phospholipase means it's going to do something. It's going to catalyze some effect on a phosphorylated lipid. And the substrate is the same one. And it's a very different signal transduction pathway. So I'll just go back to this one and this new molecule, phosphatidylinositol that you can call PIP2, means it has 2 phosphates. PIP3 has 3 phosphates. Gets phosphorylated there. This molecule can be phosphorylated on the PI3 kinase signaling pathway. In a totally different pathway, what can happen to it is it gets cut right there. And the tails stay in the membrane and become a signaling molecule, and the head floats out into the cell and becomes a signaling molecule. So I did that intentionally, to talk about 3 different pathways, and they're distinct, but the second and third pathway share a common target molecule that they are modifying. And the first one just phosphorylates it, and that's got a great name for a phosphorylating group because it's called phosphokinase C, like that. And this cuts it, right? So it's helpful to make it a little challenging, but also it comes apart easily because you're like, yeah, that's what it does. And this used to be in our textbook, and they just don't include this figure anymore, like that. So this is one where Spencer might want to take a picture of this one because it's not in our book, but also we can talk about it, and I'll also draw out what happens because it's a pretty simple story, and that is here's a receptor. It's a receptor tyrosine kinase. It gets bound to its ligand and activated, so it undergoes this autophosphorylation, and the phosphorylated form binds to this phospholipase C. PLC is phospholipase C, and it cuts PIP2. Like I said, It's an enzyme. And see how it's anchored in the membrane, right? And so now the disco ball is going to get released and come down and roll around. And the tails stay in the membrane. And both of them become signaling molecules. So we get, 2 for the price of 1. Okay? And the thing that stays in the membrane is called, and we'll draw this out, so we'll just kind of help you see it better. So for our third pathway, that's the third one we'll learn in our course. For a third pathway, we're going to have a receptor tyrosine kinase. And it binds to a ligate, so it gets autophosphorylated like this, and then it binds to this enzyme, phospholipase C. And it cuts this phospholipid into 2 parts. So it cleaves that into 2 parts, and now we just have these tails, and the tails are stuck in the membrane, but they can float side to side and activate things. And it has a longer name that I'll save, but it's called diacylglycerol, and people like to call it DAG. And then this head is very hydrophilic, so it gets cut and floats away all around inside the cell. And this, I think mostly we call this IP2. Maybe call it PIP2. Let's just see what book uses, so we make sure we stay consistent with that. Yeah, and it's IP3, so we'll call it IP3 based on that. There's some different stories that happen there. So we get 2 signaling molecules for the price of 1, DAG and IP3. Okay. And the DAG is going to stick up here, and it's going to activate. So we'll go back to our drawing, and it's going to most famously get bound by a different protein called protein kinase C, and just activate it. And we are not going to learn, I don't think in our whole course, I don't think we'll learn anything more about that pathway. There's lots, we could talk about. We will learn about this IP2 and what it does. And so I'm just going to erase this and say that now inside this cell, we have these IP2 molecules binding, moving around and binding and activating things. And the one thing we're going to especially focus on is inside the cell, there are organelles. And our example will be the endoplasmic reticulum. And in a variety of different organelles, what happens in cells is they can store calcium, and they can increase the calcium concentration, like that. So we have a cell like this, and this IP2 can bind to a receptor in the membrane of this organelle and cause the calcium to be released into the cell. And so that's going to increase calcium concentrations in the cytoplasm. And we probably also aren't going to learn too much about that, but that's a next step in that pathway. And we want to learn about that because we just have seen 3 small molecules on this phospholipase C pathway that become little molecules that are signaling molecules inside the cell. And there's a good chance at some point in intro bio you learn this term, second messengers. And maybe you learn it specifically in certain classes, like maybe human anatomy and physiology, maybe you learn a little bit about second messenger. And second messengers are a group of small molecules that are all involved in these signal transduction pathways. And so on the list of second messengers, these are 3 really famous second messengers: calcium, IP2, and DAG. So I'm just going to show you that. So here's just a summary of second messengers are small molecules or ions that release signals from cell surface receptors to target molecules inside the cell, greatly amplifying the original. Yep.
Sorry, is it IP2 or IP3?
Oh, so it could be either one.
Okay.
Either answer is good. The book in the old thing called it IP3, that's fine. Sometimes it's IP2. But yeah, so either one is good. So here's a list, and we don't need to learn them all. I just want to show you that on this list we have some famous ones. Another one we're going to meet this morning is cyclic AMP, but not these others. So it's not a list we're trying to memorize, just that DAG and IP3 and calcium are second messengers. And so see, here's DAG, here's IP3. That PIP3 is considered a second messenger in the phosphokinase C pathway, right? So we are learning about some of these famous second messengers, and there's calcium like that, right? So in that list, we're not going to learn the other ones at all, probably, but we got a nice list, a question on a test is name, 2 points, name 2 second messengers and describe how they get activated, something like that, right? So because this is incredibly important, like, it comes up in introvile, so, that must be really important, and it is, yep. So again, and one of the things is, why is that not moving? Oh, just stuck, I guess, okay, yeah. And so that's what we want to do with this list, and I'll just go back to that, and we're done with this list. We're going to, have this assignment about RAS. That's our big one that we're learning great detail all about it, and the, ALK, phosphorylated ALK, and interacting with SRF. And these, we didn't even complete the story of what they do to the cell, but we just want to demonstrate that the receptor tyrosine kinases can work through different signal transduction pathways. Yep. Okay. And, yep. So this just shows that idea that this IP2 or IP3 is going to bind to a channel receptor in one of the inner organelle membranes and lead to a release of, they call them calcium stores of calcium, stored calcium inside organelles, just being collected there to release a signal like that. And super big deal in all animals, very common pathway that occurs through a variety of signals. This is one, by getting IP2, IP3 to come in and bind that receptor and release some calcium into the cytosol. And calcium is important for our bones. This is not about the normal role of calcium. This is just, it's an ion in the cell, and if we change the concentration, we could use it as a signaling molecule in any of a variety of cells for any of a variety of reasons, okay? So we can answer questions, we can loop back through these receptor tyrosine kinases, but we've talked about them a lot to build a framework of stuff that we need to know about them. And then we'll go on to this G protein coupled receptor family. And in its people would say, oh, it's way more. This protein receptor, this receptor pathway is just way more famous, and not famous, way more significant in our body. This pathway includes how we see, how we smell, and hundreds of different receptors in this G protein coupled receptor pathway, okay? And they get bundled together with all the different things they do. They get bundled together, these 100 different receptors, because the receptors all look similar. They're not a gene family. And it's probably because they just evolved to become so different that they just don't look related. So we wouldn't call these paralogs. They just have a vaguely similar look. And I'll just show you that look here. So these G protein coupled receptors are also called, and it's no big deal, it's kind of a little bit of the history of their discovery, and I'll explain that. This is what the G protein coupled receptors all look like. It's a very simple thing, and they're called 7-pass receptors, because every single one of them has the primary amino acid sequence cross the cell membrane 7 times. And they're just very, very common. So they evolved, evolved a long time ago and just is used again and again and again. So 7-pass, and that's why the book is showing this. Why, who cares, right? Who cares? And the answer is just simply this, that these, when a protein is crossing the membrane, it's a transmembrane protein and it has to cross the membrane, have amino acids cross it. Those amino acids are always really hydrophobic, amino acid side chains, and there are about 20 of them. And so these proteins all have 7 patches of 20 amino acids. And as a result, 25 years ago, when people were starting to get a lot of DNA sequence, like the human genome, they were able to search like for this pattern. You know, show me a reading frame, a coding reading frame in the human genome that has 7 patches of 20 hydrophobic amino acids, like that. So like our book focuses on these 2 scientists that discovered the olfactory receptors in humans and won a Nobel Prize. And they found those by just looking and saying, we probably have a lot of olfactory receptors. We have like 600 different olfactory receptors because we're mammals and mammals smell really well. Like that. We don't smell as well as a dog, but all mammals have a pretty good sense of smell. And let's see if I just have a picture of them. I don't, but that's okay because I'm going to tell you this funny story anyway. I lost my place a little bit. And we're not going to talk about the olfactory receptors, but that's why people still talk about, oh, there are 7 pass membrane receptors, because they found them all really fast. And they were just, we don't know what these are. We got to figure out what all these do, you know, like that. There are many, many different ones like that. And they do a lot of different stuff. It's how we see, it's how we smell, but just all kinds of different parts of our body. So the 2 scientists that discovered the olfactory receptors were like a professor at Columbia and his postdoc student, and who, I can't remember her name really well, but she won a Nobel Prize, but did not go on to be a famous scientist. Her last name I think might be Buck. And anyway, it was 15 years ago, and at the USM Biology Club, there was a time when it was Charles Darwin's 200th birthday, and there were Darwin Day celebrations around the world. And so the USM Biology Club had a cake contest for decorating cakes in the theme of evolution for the whole university on Charles Darwin's birthday, which is coming up. Like it's right around, it might be today. Oh shoot, we should party. Yeah, and he's like 212 now, right? So, and also Abraham Lincoln's birthday, so it's right around President's Day. Okay, anyway, so the USM Biology Club was like, that Darwin Day celebration really really great. We should celebrate another scientist, just pick one. And so the Bio Club had a meeting, and a student, Jasmine, is supposed to look up the names. And there's already Google and Wikipedia and stuff. And she said, and she is like a computer science minor. It's really confusing why she said this, but she said, it turns out it's really hard to find birthdays of famous scientists. And you're just like, really? But she found this woman's name who had, she won a Nobel Prize, but not a famous scientist, and she worked somewhere. And so the USM Biology Club found out what her birthday was and celebrated her birthday, and they called her up on the phone that day. And it was just really great because that scientist was really surprised. And the students had a really hard time explaining to her why they thought she was really great. So I always remember that. Just the olfactory receptors are G protein coupled receptors, and those 2 people discovered them, and USM students had a great time celebrating those 2 scientists. Yeah. So what we're gonna do is we're gonna learn about 2 of these hundreds of receptors, and we're just gonna learn about 2 just very specific examples. Our book has a bigger section on this than on the receptor tyrosine kinases. It's a big deal. So this is a place where there's a good chance there might be a couple of questions on the exam about G protein coupled receptors that we didn't even talk about in class, right? Because there are going to be some questions like that. We just don't have enough time. What I'm doing is introducing these things in class, and that's the most important thing, to get a framework. But what about, the important other stuff the book covers? So we're going to talk about 2 categories of, because they're families, of G protein coupled receptors, and they're the adrenergic receptors and the dopamine receptors. And again, they're just super famous family, okay? So adrenergic receptors are receptors for a variety of molecules, and they do a variety of different things. And we're going to focus on one of the signaling molecules that activates an adrenergic receptor. And the molecule is epinephrine, and that's the modern name for adrenaline. And this G protein coupled receptor gets activated when a tiger walks into the room, and we freak out. So that fight or flight response is through this receptor, the adrenaline, now called epinephrine signaling pathway, and it's going to activate it, okay? So the next thing to say is we pick this partly because, oh yeah, you know what that is, and you know the feeling of the response. Because when we talk about the response, the cellular response, you'll be like, oh, I get that, satisfying, right? So so we have to kind of back up and explain a couple of facts of life that fit into this example. And so one of them is this idea of, well, the cells are going to make a response, right? This epinephrine is floating around and binding to receptors on cells, and they're going to respond. And the cells we'll focus on, as our example, we'll think about muscles, right? Because we're going to use our muscles to get the F out of here, right? It's a tiger, right? It's not somebody's dog, it's a tiger. We're out the window, right? I do not want to be eaten by a tiger, you know? Like I'd be pushing you down in front of me and be like, I'm out this window. I don't care if it's closed. Yeah, like that, okay? So one of the things is that is it going to be a genomic response? Are those muscles going to make a genomic response or a non-genomic response? And the answer to that is genomic responses take time because we're going to activate new gene expression. So it would take a couple of minutes, really, for a genomic response. Like that heat shock response, it actually takes a couple minutes to start making those important proteins. It takes a little while, too long. So the first thing we'd say is, oh, that fight or flight response, that's a non-genomic response, you know? And it's a good one for us because you're like, it better be. I got to get out of here, you know, like that. And then, so, you know, we said, you know, these cellular responses can either be genomic or non-genomic or both. But this would be an example where, you know, it's going to be a really famous non-genomic response. Then the next thing is this. So you know what our muscles are going to need is a bunch of sugar, right? And you've eaten sugar and you know how a sugar rush feels, right? If you get exposed to a bunch of sugar, you're just like, whoa, you know, like that, right? So what's going to happen here is that we're going to get a bunch of sugar for the muscles to burn right now. The sugar is already in the muscles, and it's stored as a molecule called glycogen. And glycogen is just a polymer of glucose. And it's just stored in our muscles right now. We have a big reserve of glycogen in just about every one of our cells. And it's there for a variety of reasons, but it's there for this reason. And so the non-genomic response is the receptor is going to get activated, and we're going to activate an enzyme that just cuts that glycogen up. And we have a sugar rush in our muscles right now. You know, the sugar rush you're used to is you eat it in a couple minutes, you start feeling high. This is now, and it's massive amounts of sugar for your muscles to burn, like that, right away, okay? So that's a good one to focus in on, right? It has a lot of good relevance. And it looks like this. So we first have to introduce the G protein coupled receptor pathway, like that, and then find out the enzyme, you know, that breaks down the glycogen. So all G protein coupled receptors are called G protein coupled receptors because the first thing they do is they activate a G protein. And we already remember we said this is called, people don't often use this name, but it's a trimeric G protein. So RAS is a monomeric G protein. And I'll just show you here, right? RAS is involved in receptor tyrosine kinases. The monomeric RAS plays a role up here, and these trimeric G proteins play a role down here. So we need to make that distinction really clearly. Monomeric G protein, part of the receptor tyrosine kinase signal transduction pathway. Trimeric G protein, part of the G protein coupled receptor pathway. That's why they're called G proteins. And this is what their activation looks like. There are 3 different subunits of a trimeric G protein. we're not gonna worry about that. What we're just gonna emphasize is that, well, they're called G proteins because they are always bound to either GDP or GTP. And when they get activated, when they're inactive, they're bound to GDP, and when they're active, they are bound to GTP. And so the first step in their activation is exchanging GDP for GTP, and then they're anchored in the membrane, they float around, and they're going to bind and activate other target molecule. So it's a G protein, it's got that same story like that. Remember, we emphasize we really care about RAS in our course because it could cause cancer. These pathways generally aren't on growth regulation pathways, so not gonna, mutations in the trimeric G proteins are not gonna really lead to big stories in cancer biology. They're more about everyday life, like getting out of the way of a tiger and seeing and thinking, or whatever. Yeah, thinking. Yep. Okay, so that's always the G protein coupled receptor pathway is going to activate G protein by trimeric G protein by this exchange of GDP for GTP, and then as long as it's bound to GTP, it's active. It's going go, 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 and we're amplifying the signal. And for us, we're just going to learn one target of all the G proteins, one target, and it's a famous one, and it's called adenylyl cyclase. And when it's active, it takes ATP and it catalyzes a conversion of it to make cyclic AMP. So, activated G protein receptor activates this trimeric G protein, activates adenylyl cyclase. It's an enzyme that catalyzes ATP conversion to this cyclic AMP, CAMP. And CAMP is another famous second messenger. So I'll just scroll back to that list to see that, to show that. First one they want to talk about, cyclic AMP. So that's what we're learning about. The fight or flight response involves adrenaline, epinephrine activating a G protein coupled receptor, activating a trimeric G protein by exchanging GTP for GTP, activating the enzyme adenylyl cyclase, and then catalyzing just a massive conversion of ATP to cyclic AMP. Here's a good example also. We think of ATP as playing a role in energy, right? Here's it playing a role in information. We can make a second messenger really fast with some of the ATP in the cell. So we do, and get that going, and then, and that's a figure from your book just showing that conversion, that activation of the G proteins like that. And then a little thing about epinephrine, adrenaline triggers that rapid breakdown of glycogen to glucose and liver and muscles, and it's that fight or flight response, or big part of the fight or flight response, right? We also get a big rush in our head too to be thinking about whether we want to go out the window or not. So here's just the rest of that pathway. We started out here just showing the general signal transduction pathway, and then sometimes it activates this. We get cyclic AMP, and we activate cyclic AMP binds to and activates an enzyme called protein kinase A. And protein kinase A activates this, where's the enzyme's name? I forgot the enzyme's name. The enzyme that actually cleaves the, I'll find it. But we're going to phosphorylate and activate the glycogen cleaving enzyme. It's on here. I guess it's not. Sorry. So I'll get the name of that, because we want to know the name of that, because then we're done, right? We're just going to cleave that glycogen up. There's the sugar. Go for it. And it's in the muscle cells, right? It's not going to get out in the blood and get picked up by, like that. It's going like that. Yep. Questions?
Can you say one more time, the activated G protein, does it bind to the adenylyl cyclase? Yeah. And then can you explain what happens right after that again?
Yeah. So as long as the G protein is bound to GTP, it's active. Mm-hmm. And so this active G protein will bind to the adenylyl cyclase, And as long as it's active, the adenylyl cyclase is active, and it's catalyzing the reaction of ATP to cyclic AMP. And then there's this additional step of, all that big amount of cyclic AMP is floating around, and it's activating another enzyme, protein kinase A, that's phosphorylating another enzyme, and we don't need to know them. And then it's going to activate phosphorylate that target enzyme that does the job, that cuts the glycogen down to glucose. And I just can't remember the name of that right now. Like that. And that's that story. Yeah, Chase?
You said we didn't need to know the rest of the pathway.
Well, so, it's pretty complete here, right? So epinephrine, this adrenergic G protein coupled receptor pathway receptor, and then the G protein, trimeric G protein, then the adenylyl cyclase, ATP to AMP, and then cyclic AMP, cyclic AMP binds to protein kinase A. There's this additional kinase that's written here, but we don't need to know the name of it. It's just, amplifying the step. And we do need to know the name of that final enzyme. I got to get that. And the substrate is glycogen, and it cleaves it to glucose. So we need to know the pathway except that one kinase name, and I got to get the name of the final enzyme that's activated. And that all happens really fast. And what is this? On an exam, say, give one example of a genomic response, and it would be like, well, RAS. That RAS pathway is a genomic response because it's leaning into activating gene expression. What's an example of a non-genomic response? Oh man, fight or flight. And this whole pathway, draw it out, explain it like that, explain a little bit briefly, because it's just really satisfying, right? Molecular biology is always really satisfying. It gets complicated, it gets a lot of pieces and stuff and names I don't remember, but it just is like, oh my gosh. Is there more to it than that? The answer is no. When your muscles get activated by a big sugar rush, you're wiggling and, getting out of here, right? Like it's just a whole bunch of sugar, right? Like that. In the cells, in the target cells, like that. Okay? And yeah, I'll get that, for next week, like that, okay? So that's our first example of hundreds of examples of G protein coupled receptors. That's a good one. Adrenergic receptor role in the fight or flight response. Adrenergic receptors are a family. They play a lot of different roles, but that's just a really sweet, clean one for us to walk around and feel, right? When I say boo, you go, whoa, man, my adrenergic receptors are freaking out, right? And I'll just say as a side note, I don't know, maybe it shouldn't be on this, but when the police come through, or the safety officers come through to lock up the building, it's like at about 9 p.m. and I'm off in there working and stuff like that. And one time I was coming out of a lab, and I was like, I got to get out of here, I hate my job. And I slammed the door, and then there were 2 doors on this classroom, and I slammed this door, and I slammed this door. And I came rocking really fast around the corner, and that safety officer was like that. I couldn't draw. Like there's shots being fired in the science building. I was like, oh my God. Don't hurt me. Yeah, no, we were both having serious fight or flight responses. That was really scary. All right, second one, dopamine receptor. It's in our head. It's a neurotransmitter. It's a really good example that'll just freak you out to hear about it because you'll just be like, oh my gosh, molecular biology is the coolest. And why don't we talk more about the bodily actions of all these little molecules? Because dopamine is really great, and there's a really good chance you've heard of dopamine. It's a famous neurotransmitter in our brain, and it's involved in the pleasure response. So sometimes you say, oh, I got an endorphin rush, right? I was out there doing this or that, skiing or whatever I really love to do, and I got this big endorphin rush. It turns out dopamine is also involved in that pleasure response thing, and it's a neurotransmitter going between neurons. And this is not a neurobiology class. We're not going to dive into that at all, but just raise your hand if you took neurobiology already here. So we have some students like that. And so, it's not going to help you on the exam, but it will help you a little bit here. Because what we have here is this image, and this is called a synapse, and it's just a little gap between 2 neurons in any nervous system on our planet. So we'll pick humans. And here are just the terms we want to mention. So here's what we would call a source cell, and every neurotransmitter is one of these signaling molecules that we're learning about. And then there's the target cell. So these are just like the kinds of signaling factors we're learning about. They're just very specialized because this synapse is just a really little tiny gap. It's all very isolated. They're not releasing it to float away in the blood or anything like that, right? These aren't hormones, they're paracrine factors, and they play this really restricted, focused role, like that. And every time we think or move, we're releasing a lot of these neurotransmitters because they carry the action potential signal, that electrical signal, from this neuron to the next neuron. It has to jump across that little gap of that extracellular space, okay? And so dopamine would be a neurotransmitter, and it would bind to a receptor here. So that's what we're talking about when we're talking about dopamine. We're talking about a neurotransmitter, and it's going to be binding to a receptor, and it's called the dopamine receptor on the surface of another neuron. Like that? Like that, okay? And so there's going to be a cellular response here. When we're thinking, that's happening really fast. So do you think that the response to neurotransmitters would be a genomic response or a non-genomic response? Yeah, big time. It takes minutes for a genomic response, and we're thinking in microseconds, right? It's just big time. And what happens here is these receptors are called channel receptors, and they just let ions in and out. And that's what the action potential is just a change in membrane polarity and an electrochemical gradient across that cell membrane. It just happens like that. So there's dopamine receptors that are channels, and they get activated when we feel good. Dopamine plays more roles in our brain than that, but that's the one we'll focus on, is that feeling good. That endorphin rush also includes not just endorphins, which are neurotransmitters, but this dopamine, okay? All right, and that's, the receptor is a channel protein, not a G protein, coupled receptor. Those don't have channels to let ions in and out. This is a different kind of receptor. The channel just opens a pore, opens an opening, and lets in and out certain ions. Sodium, calcium, so forth, like that, okay? So our receptor is not shown here, but it's on the surface of this cell, and it is a G protein coupled receptor that's a dopamine receptor. So in the story of dopamine, there are two different categories of receptor. The channel, which brings that signal, the thinking signal, the thought, very quickly through a non-genomic pathway, just in the cell membrane, just changing the resting potential of the cell membrane like that, letting ions in and out. And dopamine also binds to a G protein coupled receptor called the dopamine receptor. And that's what we're focused on because it's a G protein coupled receptor, and it's going to cause a genomic response in this cell. We're going to have some new gene expression, okay? So I'm just going to go back. This means my little thing is running out of power, but I probably won't have to draw anymore here because we'll be done with class. Because we'll just keep going. Oh, we have plenty of time. Let me just plug this in so it starts charging it. Yeah, and this example is going to mess you up. That's a very good example. So before we dive into it any further, I'm just going to go back and just say, right, that first one we learned about, so the fight or flight response, we got the G protein activated. It activated adenylyl cyclase, and it activated protein kinase A. G protein coupled receptors have lots of different things that the G protein activates. But I intentionally picked these two examples because in this nerve cell, this neuron, the dopamine, just to show you that we have a non-genomic response here. The dopamine is causing a non-genomic response. And here it is from our textbook. The dopamine can also cause a genomic response in the same cell. So there's going to be the genome, and it's going to activate gene expression. Right? Okay? And the first part of it looks the same as our fight or flight story. The G protein gets coupled. Who's binding to it? In this case, it's dopamine. And it's a neuron, activates this G protein. Again, activates that adenylyl cyclase, but it's not a muscle, it's a brain cell. And in this case, activates protein kinase A again, but it's a brain cell. What's going to happen? And the answer is part of this protein kinase A actually floats in and phosphorylates a transcription factor and activates that transcription factor. Yeah? And the transcription factor is called CREB. And that's just a great name. And great names usually come from biochemists, not geneticists. And so we're going to see, we can just draw this out. So dopamine is going to bind to this receptor. That's a 7-pass receptor, a G protein coupled receptor in a neuron. It's going to activate this trimeric G protein, which is going to float around anchored in that inner part of the cell membrane, bound now to GTP. That trimeric G protein coupler is going to bind to adenylyl cyclase, which is an enzyme that's going to catalyze the conversion of ATP to cyclic AMP, which is a famous second messenger. And cyclic AMP is going to bind to protein kinase A. And our book shows this pathway. Our book is not saying it's dopamine, because this same pathway is used by some other ligands and receptors in other settings, but I'll get that figure number and tell you that's our dopamine receptor figure. And then this protein kinase A is going to float into the nucleus and phosphorylate this protein that functions as a dimer. So phosphorylated form is an active transcription factor, and it's called CREB. And you remember last time we met, we ended class with the serum response factor, and it's really satisfying to understand why they called it that. So CREB is also the same story. It wasn't in this dopamine receptor example. CREB is activated in a variety of different settings, and what CREB stands for is the cyclic AMP response element binding protein, right? Because scientists found a way where cyclic AMP levels increased in the cell and something happened. It wasn't this dopamine story, another cell. And something happened, a cellular response happened. And they're like, I wonder if it's a genomic response. Let's see if there are new genes that get expressed when we increase the cyclic AMP. And then there were, and they're like, then we're going to call this a cyclic AMP target gene. And we're going to try to find the enhancer by doing some deletion studies and some nuclease footprinting and some gel mobility shift stuff like that. And we'll call that enhancer the cyclic AMP response element. So lots of people, when they get specific, they don't call it an enhancer. It's an enhancer, but they call it a cyclic AMP response element. It's what's responsible. It's the part of the cis regulatory region that's responsible for causing this to be a gene that's a target gene for cyclic AMP. And they're like, what the heck is the transcription binds to that, you know? And the graduate student working on the project says, I know what I want to call it. I want to call it cyclic AMP response element binding protein. And that's CREB. So it's a sweet, sweet story, right? Like that. Okay? All right? And then we're not going to name the function of any of the target genes. But, back to this. When dopamine makes this non-genomic response through these channel receptors in the neuron, you feel good. At the same time, dopamine is using our pathway, the G protein coupler receptor, dopamine receptor, to change gene expression. And what is happening in the brain when that's occurring. So it's a longer term response to dopamine stimulation. Like that, okay? All right, and so we're wrapping up today on this, and we have a few more minutes, so we're doing great. Hi, we're doing great. All right, so here's the story, right? And so this is the bottom line of why this is super crazy. The relationship between dopamine and the cyclic AMP response element binding protein, CREB, is a critical molecular mechanism in the development of drug addiction. Like that, right? I feel good. I'm stuck on feeling good on this gambling or this drug and, addictions, right? The CREB is a critical molecular mechanism in the development of drug addiction, acting as a bridge between immediate drug-induced pleasure, the non-genomic channel response, and long-term neuroadaptations. Right? The person develops this addiction over time, and then it really locked in on it. Well, dopamine causes initial euphoria. Chronic activation of dopamine D1 receptors leads to the activation of CREB in the nucleus and a certain area of the brain, which contributes to tolerance, dependence, and negative emotional states of withdrawal. Super messed up, right? A little bit, every once in a while, great. A lot, frequently, and it really changes the person's brain, right? And getting out of drug addiction means getting out of the changed gene expression in the brain that's a cause. This isn't the only story. We're not oversimplifying, though. This is a big part of the story of addiction. Like that, right? So just a beautiful example of us to tie in, a bunch of these things. If this is G protein coupled receptors, the book spends a lot of time on them because it's massively important. And here are two examples of G protein coupled receptors where you're like, oh my gosh, let's go get tattoos right now. Class tattoo, right now. And it's crab. You just be a crab on your back and be like, is that supposed to say crab? And you're like, I don't know. I hope not. Like that, okay? So that, is all I really need to say about that section. There's more to it, but, it was a basic framework of G protein receptors, and we have two really good examples that we can talk about. And maybe, you can learn about some other examples because the book talks about, the pathway in vision. But it gets really, fussy and not so much like you're like, oh man, this is good. These are good examples. Yep.
So is the difference between the two, once you're at the protein kinase A, the phosphorylation, you either get CREB for the dopamine receptor or the enzyme, and the other, that's the difference?
Yeah, so I picked that intentionally because the pathway looks really similar, but it's in a different cell, right? So the first example for the epinephrine is in muscles. It's also in liver and in some other parts of the body for that fight or flight. And here the dopamine story is in the brain, in nerve cells, neurons like that, right? And so it's a different context. And so that's part of the story of these signal transduction pathways is they have, they have like a different biochemical cellular environment. So they're set up to, finish up that pathway with different, steps like that. Yeah, Liana?
You said we are going to have to look in the book for the exam as well. Are we going to need to know any additional examples of receptors other than the ones listed?
I hope so. Yeah. And so, I say, you got to get this book and you got to read it. And I can't talk about all of it. And so if you have questions, bring them into class. Yeah. I would be asking a lot of questions about the book. However, I also tell every class I teach, I say, I think if a student really understood what was going on in class, they would get a B. The only way to really understand it is also to find those parts in the book, and it's not the whole book, So there'll be some questions, but not tons of questions that are from the book that are not in class. Yeah?
You said that the book talks about a lot of G protein coupled receptors. So are there other specific ones we didn't go over that in class too?
Just the ones in the book, yeah. And they won't be like, tell me the whole pathway, but it might be, say, give examples of different settings in our body where a G protein coupled receptor is regulating activity. And I'd mentioned vision, the book talks about it and shows that. Smell, it's got those two scientists in a box, like that. So they're not trick questions or anything like that. But, it's like that where, and the way I describe this to classes is, so the deal with biology is that it's too much information. So if you stopped and asked a biologist directions of like how to get to the mall, the biologist would say, oh great, I'm going to give you 3 completely different ways of getting to the mall. Not different routes, but just different stories. The first one is going to be using street names and north, south, east, west directions. The second one is going to involve landmarks, like turn right at the McDonald's, turn left at the big red barn. And the third one is going to provide examples from my life. I got engaged at that place on that corner. I got in a crash. My mom got in a crash there, like that. And you're just like, quit it, right? But that's what makes biology tough, right? Charles Darwin just rode around the whole world on a boat, looking at all of the animals and plants, all of them, and being like, I got a lot of examples. We might sink the boat, there's so many examples. Yeah, and so we gotta practice that, right? So that's how I think of the book, is we gotta practice struggling, because you don't want to erase your class learning by looking at more examples in the book, which happens, right? Start looking at more examples and you're like, dang it, I lost track of that epinephrine story so bad, right? But it's just practicing it. And it won't be tricks and it won't be a lot of points. Definitely, after the first exam, students won't have done as well as they want to, and I'll say, well, really focus on the class. That's, like that. That's to record it, man, go back and watch it again immediately, like that. Yep. Yeah. Mm-hmm. Yeah, and I'm kind of smiling and laughing just because I'm a biologist, and biologists all think that's funny, where they just go, yeah, I can tell you how to get to the mall. Just give me like 20 minutes, because it's so cool. Because I get to talk about my engagement. What was so cool, like that, right? And you're just like, oh, we're driving away from you, like that, right? So yeah, that's the role of the book, yep. And, and I'm not trying to dodge the book. These are, examples that are in the book generally, and stuff like that, yep. And we got to cut out. But bring those kind of questions to class, right? Because next time we meet, it'll be a week from our first exam. Yay! And before I turn the recording off, molecular biologists don't care at all about any of these fight or flight responses or drug addiction type things. They're just like, look at that receptor go, man. Chase, do you have a question?
Yeah.
Yeah.
Exam's on the 26th.
Oh, it's on Tuesday, I think.
Tuesday.
It's in our syllabus.
Oh. For some reason, I thought this class was on Monday next week, but it's on Tuesday. So
Yeah,
I was
and Monday's a holiday.
Yeah, that's why I was confused.
Oh, yeah.